Okay, this is the third session from Dr. Neil Cross on Biomedical Investigations, and we're going to continue the theme of looking at genetic analysis. And in today's sessions, we're going to look at two or three different mechanisms of looking at genetic mutations for cancer subtyping, and that is multiplex ligation dependent probe amplification and single nucleotide polymorphism stroke mutation analysis. So what I'm going to cover in this molecular diagnostics session, I'm going to rely upon you understanding PCR. I know you've covered PCR in detail in previous sessions. You've covered allele-specific PCR, qPCR, real-time PCR was certainly covered last week. And what we want to do is build upon your knowledge of these and apply them to other clinical applications, because we can use some of these concepts to build on this for a more complicated method. Now, the first method I'm going to talk about is multiplex ligase dependent probe amplification, which requires PCR concepts to be understood to understand how this works. So I'm going to talk about this first, and then in the second half of the session I'm going to talk about single nucleotide polymorphism stroke mutation analysis, and then how we can do this using quantitative PCR principles. So the first technique that I'm talking about is NLPAs. And this is a technique that is used for the detection of chromosomal losses and gains, or losses and gains of individual genes, or losses or gains of individual exons within genes. So we use this assay if we want to simultaneously detect up to about 40 different targets for gene copy number or DNA copy number. And it's dependent upon what is quite an old technique called the ligase chain reaction, which was a technique that was developed in popularity, but PCR really took over from ligase chain reaction. First, I'll tell you about the ligase chain reaction, and then we can look at how the first part of a ligase chain reaction is used in an MLPA. So ligase chain reaction, it's an alternative to PCR, useful for some applications. And basically how it works is a DNA product is created by ligating two probes together. And if you are interested in a particular region of DNA, you generate two probes, allow them to bind to that target DNA and add DNA ligase, and that will stick those two pieces together, because ligase will only work with probes which are immobilized on a template. So it only occurs if the correct template is present. If the probes are floating around in solution, they won't get stuck together. So it doesn't use any DNA polymerase, it doesn't use DNTPs. And then we can repeat this cycle with two more anti-probes and repeat this to amplify DNA so that you get the same sort of DNA amplification in PCR, but you don't use any DNTPs or DNA polymerase. So this is shown here. Here's some this is some template DNA that we want to detect. And to detect it, firstly we denature the DNA, and then we add in two individual probe sets. One which is antisense to the top strand, one which is antisense to the bottom strand. And what you can see here is I've deliberately left out a little dash there because that shows that there is no phosphodiester bond between this A and this G, and no phosphodiester bond between this C and this T. So the probes will stick to the, or hybridize I should say, the probes will hybridize to the template here, the probes will hybridize to the template here, and then we add in, once they're bound, DNA ligase, and DNA ligase will put in the phosphodiester bond here, and this will create two new synthetic pieces of DNA. Now, for the purpose of this illustration, I've done the probes as only being 5 or 6 nucleotides in length. In reality, they can be 30, 40, 50. They tend to have a recognition site of 15 to 20 nucleotides in length, but then you can have extra bits on the end, and we'll find out why later on. But the key point is, once you've added the ligase, you've created an exact copy of this DNA. So these probes here have created an exact copy of the bottom strand of DNA. So this goes CATGAG, this goes CATGAG, and so on. And all we do is then denature this and then allow the probes to bind to the other probes. So just repeat the process again and again. So you end up with these red probes, this red probe binding to this blue DNA and this green binding to this DNA. So you can design it so that you have 4 individual probes, they get ligated together into 2 single-strand pieces of DNA, and then they act as template for subsequent reactions by heating and cooling. And that is the ligase chain reaction, and you repeat that 30 or 40 times to amplify your DNA. So this is a non-PCR based amplification. It's highly quantifiable. It just isn't used very much because PCR is much more, it's just much more widely used, and there's many different applications. Now the ligase chain reaction principle is used in the first step of multiplex ligation dependent probe amplification, and it's only the first step that is used. So what we are doing in MLPAs is performing that first ligation step with 40 or more probe sets. So and then we're not doing the subsequent rounds of amplification, and I'll illustrate this on the next slides. What we then do is detect those 40 plus probe pairs. So we've got a pair of probes. So whereas in ligase chain reaction we have 4 probes, these 2 and these 2, in the first step of an MLPA we just have these 2 binding. And then we use the binding of these 2 and detect that DNA using polymerase chain reaction. So in the first step we've ligated our 2 probes together onto our template. They will only get ligated if the template is present, and then we detect all 40 of them with the same set of PCR primers, and we detect them on the basis that these ligated probes all have different lengths, and that allows us to amplify all 40 ligated products with the same pair of PCR primers, giving rise to 40 different PCR product lengths, which we can then analyse by some sort of separation method. And this is shown here. So this is just for 2 targets. So here is our first part of the probe. It's going to bind to target A, and the second part of the probe is also going to bind to target A, and this gap between them is a phosphodiester bond. And as you can imagine, DNA ligase can't link these together unless they are bound to the correct target. So this probe binds to target A, this probe binds to target B, DNA ligase comes along and sticks them together, and that creates a new piece of DNA which we can now detect. But first we need to amplify it. Now the thing is about this technique is these bits on the end do not bind to the target DNA, but they are part of the probe. These just flap around for now, and we can use these for detection purposes. Now the key part about these bits that don't bind to the target is they contain a primer binding site. So this here is a forward primer binding site, and this bit here on the end is a reverse primer binding site, and then in between we have a stuffer sequence which is different for each probe pair, so that the distance from here to here differs for every probe pair. And then when we amplify this DNA, copy this DNA, then amplify it, we get different PCR product lengths, but using only a single pair of primers. So for example, target A, when it's ligated onto the target, creates a new piece of DNA that's 130 base pairs in length, whereas target B, which is a different gene and different regions hybridizing here, the distance from here to here is 150 base pairs. But we can amplify this DNA and this DNA with a single pair of primers, because the same primer binding site here is here, and the same primer binding site that's here is also here. Okay, so I will go through the whole schema of how this works in a moment, but the sorts of situations where we would use this is if we wanted to, for example, map large chromosomal deletions or screen for chromosome abnormalities. So we can use this. If we've got some DNA from a patient, we can look at the relative number of every single chromosome by having a probe set designed to each chromosomal arm. So we have 22 autosomes plus X plus the Y chromosome, and most of those chromosomes have got a P arm and a Q arm, but not all, so that we can have a probe set designed to every single chromosomal arm and analyse the relative amounts of each chromosome arm in a single reaction without having to karyotype, for example. So we can do this for prenatal diagnosis, so we can look for aneuploidies, so trisomy chromosome 13, 18, 21, loss or gain of X chromosome, loss or gain of Y chromosome. And we can detect it all in a single experiment and then run all the amplified products down a capillary electrophoresis, which is then can separate out all the different size products. So that's the applications of it, one of the applications, and I'll now take you through the full workflow of how it works. So as we've already seen, here's target A, here's target B, and we'll go all the way up to target Z. 26 different genes, 26 different regions. For each target we have a 2-part probe set separated when they bound to their target by a single phosphodiester bond, and if the half probe binds and the other half probe doesn't bind, we cannot get them ligated. We need this probe and this probe bound next to each other. So we allow them to hybridize, and this is usually done overnight, very long hybridization, and then we add in some DNA ligase. That DNA ligase will put a phosphodiester bond between here and here and create a single stranded piece of DNA, in this case 120 bases, and this one 150, I should say nucleotides, because it's not base pairs yet, it's nucleotides. So that'll create a piece of DNA from here to here, 150 nucleotides in length, but it's still stuck to the original template. Okay, so once we have ligated the two probes together, we are left with a single strand piece of DNA. So we'll denature that DNA and we'll be left with this newly produced single-stranded DNA. There's a primer binding site there and a reverse primer binding site there, and that's the same as there and there. So what we can now do is perform PCR amplification on all of these pieces of DNA and amplify them up to be double-stranded DNA, but we're using only one primer pair. And in the first round of PCR, a primer will bind just here. So just the reverse primer will bind, because there will be a 5, remember DNA is anti-parallel, there will be a 5' end here, a single stranded piece of DNA will bind to there, which is the reverse primer. There will be a 3' end pointing in this direction, and then DNA polymerase will extend from this 3' OH in this direction. And that will create a double-stranded piece of DNA in the first cycle, and then in subsequent cycles the forward primer will be come into play, which will bind to the piece of DNA that has copied, is the copy of this. So we'll create a new piece of DNA here, denature it, and then the forward primer will be able to copy that piece of DNA, and from this point onwards we'll be able to get standard PCR amplification and doubling of DNA every cycle. And there's a link to the MLPA manufacturers there, which is where a lot of these slides have come from, explaining the process in more detail if you want it in more detail. So what we then do is separate out all of these 40 pieces of DNA, these 40 amplicons that have all been amplified by the same pair of primers, and we do that using typically capillary electrophoresis. And so we run all these PCR products down a capillary column, separate them out by size, and we can label one of the probes with a fluorescent tag, so therefore we can see the DNA as it comes off the column and quantify that. So we can see the whole process here again. So target A, target B, the 5' end of the half probe is bound here, the 3' end is bound here. Because they're both bound perfectly to the target, DNA ligase can ligate those together to create a single stranded piece of DNA, and then a reverse primer can start to, in the PCR stage, copy this DNA so it's double stranded, and then from that point on you perform 30 or 40 rounds of PCR amplification, and you're using the same primers for every target to amplify every probe set, and then you run the whole lot down the capillary electrophoresis column, and you get peaks as pieces of DNA come off at different sizes. The smallest ones will come off first, the longer ones will come off later. Now when we look at an MLPA result, you get something like this. We always have a normal reference control. And in this situation, we're looking at the APC gene, which is the adenomatosis polyposis coli gene. It's a gene that is often partially deleted in colon cancer. So we can use it for screening deletions within a gene. So to do this, we would design MLPA assays for every single exon throughout the APC gene, which is a particularly big gene, and then look at the peak heights for a normal DNA sample. Now the peak heights differ in size due to a combination of the size of the amplicon that's produced. So a smaller amplicon might amplify more efficiently than a longer amplicon. So you can end up with different peak heights. And also you can get different efficiencies of probes binding to template DNA. So there are various factors that can affect the peak height. Also, so you end up with different peak heights, but as long as you've got a normal reference control DNA, you've got something to compare to. So you run this at the same time as a patient sample. And what we're looking at is just the peak heights. And you can see for this control, and this promoter region, the peak heights follow the normal DNA. So that's a particularly low peak for whatever reason. That's a much higher peak, that's even higher. And the pattern follows pretty much perfectly until we get to this point here. This intermediate height peak, you'd expect the next one to be a bit higher. It's not, it's about 50% lower. And what that means is that exon 11, which is where this probe set binds, there is only one copy of it. So basically one copy of the APC gene has lost exon 11, exon 12 and exon 13 because the peak heights are all about 50% of where they should be. So that's how we score it. We're just looking at peak heights and also area under the curve and using that to assign gene copy number. This is diploid, 2 copies. This is just 1 copy because exons 11, 12 and 13 have been deleted from the APC gene in this particular patient. So here's some more examples. This is using normal female DNA as a reference. If we take DNA from a male, we can see that the X chromosome peak is roughly 50% of where it is in a female, whereas if we have a triple X female, so that's 47 XXX karyotype, this peak height is higher than you would expect it to be. Now we can also use this for cancer subtyping. If we want to screen for lots and lots of different oncogene mutations or tumor suppressor gene deletions, oncogenes drive cancer growth when they're overexpressed or duplicated. Tumor suppressor genes promote tumor growth when they are lost. So what you find with tumor suppressor genes, you have 2 copies of a tumor suppressor gene, 1 on each autosome, and if you lose 1 copy, so if a cell loses 1 copy of BRCA1, that could render it more likely to become tumorigenic, and then often the second copy of the gene gets mutated. But what we're looking at here is a tumour which has lost 1 copy of the BRCA1 gene. Here we're looking at a different tumour that has gained the ERB2 gene. Now you all should be aware of the ERB2 gene. It's also known as HER2. So here is a normal reference sample DNA, and we can have a probe set for all of the genes that are relevant to breast cancer. That would be BRCA1, BRCA2, HER2, maybe epidermal growth factor, and various other genes. And what we can do in a single experiment is find out whether the main cancer-causing genes are either lost or gained in this pathological sample. Okay, so let's just look at the bottom one. We can see that the BRCA1 gene, which is represented by this one here and this particular fragment here, is 50% deleted. So that's a BRCA1 deleted cancer. Whereas in this one, you'll see the scale is different here because the signals are so high. But this peak represents the ERB2 or HER2 gene, so we know it's in a HER2 amplified cancer, and also cyclin, one of the cyclin D genes is amplified. Cyclin D is one of the things that helps control the cell cycle. So this has got cyclin D amplification and HER2 amplification. So we can test there'll be lots of other different oncogenes and tumor suppressor genes represented by various peaks on here. Okay, so that's the end of the first half of this lecture. This is on MLPA. The next video that will be coming up will be on single nucleotide polymorphism and mutation analysis.